What if I told you that some Alzheimer's drug that failed actually worked brilliantly, but only for 43% of patients? And now we have the statistical tools to identify exactly who those patients are before they even start treatment. For us APOE4 carriers, this changes everything about personalized medicine and how we approach treatment. Hi, I'm Dr. Kevin Tran. I'm a doctor of pharmacy and an APOE4 carrier. I'm also the founder of the Phoenix Community, which is a platform dedicated to helping APOE4 carriers like you and I beat the odds and outsmart Alzheimer's. Through my personal journey with APOE4, I dive deep into the latest research and translate complex findings into hopefully clear and actionable insights that we can all apply today to take control of our brain health. Today, we are exploring something revolutionary from the AAIC conference in 2025, where 6 different statistical innovations that are converging to solve the biggest problem in Alzheimer's research: the fact that clinical trials that fail not because the drugs don't work, but because we've been measuring success the wrong way. What you're about to learn could determine whether we get access to effective treatments in the next 5 years versus the next 20. And for us APOE4 carriers, these breakthroughs are particularly crucial because we often show different treatment responses than non-carriers due to our accelerated amyloid accumulation and our distinct inflammatory profiles. So once again, personalized medicine is truly the future, and all these technologies that are being invented and developed are really important for our own personalized treatments. So let me start with something that will blow your mind, hopefully. Dr. Lei Liu from Washington University just presented data showing that a major Alzheimer's trial that was declared a complete failure with essentially 0 benefits across all patients actually had profound benefits for specific subgroups. This was a massive 76-month trial with 378 patients comparing donazepam, which is a cholinesterase inhibitor that boosts acetylcholine levels in the brain, to a placebo in mild cognitive impairment. The overall result, almost 0 benefits, because the p-value wasn't even close to the typical 0.05 threshold for statistical significance. By traditional standards, this drug failed completely. But here's where it gets fascinating.
So this is a longitudinal trial, like 36 months, multi-center randomized, double-blind, placebo-controlled parallel group studies. So we have repeat measures every 6 months. And in this study, we are most interested in the comparison between donepezil versus placebo. So the sample size is 378 in these 2 arms. The outcome is a clinical dementia rating scale ranges from 0 the best to the 8 worst. And you can see the overall treatment effect is almost 0 and p-value is 0.99. So basically this is a failed trial. We cannot demonstrate the superiority of donepezil versus placebo. So a question naturally arises: can we rescue this trial by identifying some subgroups which respond to the medication?
Dr. Liu's team applied something called interaction tree analysis, essentially a machine learning algorithm that recursively partitions data to find hidden patterns where treatment interacts with baseline characteristics. Think of it as an AI that can identify patterns humans will never see by examining thousands of possible combinations simultaneously. And what they discover changes everything about how we think about these failed trials.
Okay, so you see we have subgroup 1, they have a beneficial treatment effect. MMSE is less than or equal to 24. So the sample size is 37, so about 10% of the total. So basically it has a treatment effect of 0.45. Another subgroup greater than 1.5, younger age than 75 years. They also have treatment effect 0.65. Sample size is much larger, so n equals 125. So if we combine these 2 groups together, we see we have a sample size of 162. So that's 43% of the subjects. So treatment effect 1.08. So that's more than double.
So think about what this means. Nearly half the patients in this failed trial actually had treatment effects of 1.88 on the CDR scale, which is almost double what we typically see with approved drugs like lecanemab, which showed like a 0.45 point benefit. They weren't non-responders. They were hidden responders buried in average data. The MMSE cutoff of 24 represents mild cognitive impairment, while the FAQ score above 1.5 indicates early functional difficulties, exactly where we'd expect cholinesterase inhibitors to work best. So for us as ApoE4 carriers, this is particularly relevant because we often fall into specific subgroups based on our accelerated disease progression and our earlier amyloid accumulation, and different inflammatory responses mean this statistical tool could finally identify which treatments work best for our specific genetic profile. Now let's turn to Dr. Viswanath Divanarayan from SI, who developed machine learning models using gradient boosting algorithms that can predict with stunning accuracy who will progress to Alzheimer's and who will remain stable using just baseline data. His team analyzed data from the A4 study, so the anti-amyloid treatment in asymptomatic Alzheimer's trial, which is focusing on preclinical Alzheimer's patients. These are people who have brain changes but no symptoms yet, typically with amyloid PET severe values above 1.15. This is exactly where many of us ApoE4 carriers find ourselves, especially if we are proactive about testing, since we accumulate amyloid 10 to 15 years earlier than non-carrier. And the breakthrough, a single biomarker, dominated everything else.
And you can see p-tau217 is a dominant predictor in this model, followed by some baseline cognitive assessments, along with some morphometric features in mostly medial temporal and neocortical regions.
So p-tau217, phosphorylated tau at position 217, emerged as the single most powerful predictor of cognitive decline. This biomarker, which you can get tested for around 300 to 400 at Quest Diagnostics or LabCorp, reflect tau pathology that's downstream from amyloid accumulation. So for context, p-tau217 is about 94% accurate in detecting brain changes associated with Alzheimer's, making it more reliable than many imaging tests that can cost thousands. By the way, we have an entire full video about p-tau217, so I'd suggest you go watch it on our channel if you haven't yet. The medial temporal regions mentioned include the hippocampus, the entorhinal cortex, which are areas crucial for memory formation that deteriorate early in Alzheimer's. But here's where it gets even more interesting.
So how good are these predictions? So as you can see in average profile over time, especially in the later time points when there is meaningful progression, the predictions from the two models are fairly aligned with the observed decline. And the subject level predictions are also reasonably correlated with the observed decline. We can use these continuous predictions over time to also classify individuals based on those that are stable versus those that are declining at different rates. And when we do that, we get a ROC AUC of up to 83%.
83% accuracy measured by ROC AUC, which is kind of a barbaric acronym for Receiver Operating Characteristic Area Under the Curve, in predicting who will decline versus who will remain stable. To put this into perspective, that's approaching the accuracy of amyloid PET scan, but using just blood tests and basic cognitive assessments. But the real magic happens when we use these predictions to improve clinical trials. And this is where things get really exciting for our community. If you've been following Alzheimer's research, you know one of the biggest problems is that trials require enormous number of patients, often 1,000 to 2,000 people, and these take forever to complete, sometimes 5 to 7 years. So Dr. Devanarayan's team discovered that by using their machine learning prediction as what's called prognostic covariates, essentially using the AI's prediction to mathematically account for individual variation in disease progression, well, they could achieve something truly remarkable.
So we ran simulations, about 5,000 clinical trials simulated to mimic the characteristics of the A4 data. To detect a 38% slowing decline with APC covariates, we simulated APCs with varying levels of predictive strength. So the prognostic covariates that are more predictive of progression obviously has a bigger impact on the power of the treatment effect assessment. So in particular, the model 1 that had an R-square of 32% increases the power from 80% to 90%. And the other model, which was using tau PET instead of MRI, increases the power to 95%.
Let me put this into perspective. Statistical power is the probability that a trial will detect a true treatment effect when one exists. Increasing power from 80 to 95% means trials are far more likely to detect real treatment effects. It's the difference between a drug being approved or abandoned. The R-squared value of 32% means the model explains about a third of the variation in progression, and that's actually quite strong for biological data. But even more importantly,
Now, if we lock the power at 80%, the sample size would go down. So model 1 would result in 24% reduction in sample size, whereas model 2 will provide a reduction of around 35% in sample size requirements for a future study. So in preclinical population, recruitment is very, very difficult. So on average, you need to screen around 8 to 9 people to select one person. So this kind of sample size reduction can translate to screening 1,500 fewer people. And that's a huge impact in the conduct of clinical trial.
So 1,500 fewer people needing to be screened. The screening ratio of 8:9 to 1 reflects the challenge of finding people with elevated amyloid but no symptoms. That's a sweet spot that's particularly common in APOE4 carriers. Actually, I remember a lot of our members are in this specific situation. So for us in the Phoenix community, this means trials specifically for APOE4 carriers become much more feasible. Instead of needing thousands of us to participate, we might only need a few hundreds. If you are finding this information valuable, and I know it's complex but crucial for our future, please hit that subscribe button. That's a way to support me and to support the community I'm building called the Phoenix community, to stay informed about APOE4 carriers who basically understand the science and can advocate for ourselves. Every subscription helps us reach more carriers who need this information. Now let's turn to Dr. Yan Li from the Washington University, who identified a fundamental flaw in how we've been measuring treatment success. Traditional trials look at time to first progression. Essentially, that's whether someone progresses from CDR, a score of 0, which is healthy, to CDR 0.5 or higher, which is called impaired. It's a single binary event. But Alzheimer's is a continuous journey through multiple stages, and treatments that slow progression over time might not prevent that first event, but could dramatically slow everything that comes after this first event.
However, Alzheimer's disease is a progressive disease with continuous decline. So time to first progression does not capture the full disease course. Using data as an example, the blue line is from the placebo group. The red and orange lines are from the treatment group. For the red line, slowing progression is reflected after symptom onset. For the orange line, slowing progression is reflected both before and after symptom onset. So only time to first progression, we may not reflect the long-term treatment effect.
So this is crucial for us as APOE4 carriers because we often have a longer preclinical phase, basically more amyloid buildup before the symptoms, sometimes 15 to 20 years of accumulation of amyloid. So a drug might not prevent our initial progression from CDR 0 to 0.5, but it could keep us functional at CDR 0.5 to 1 for years longer instead of progressing to CDR 2 or CDR 3. That's basically the main difference between needing personalized care at 70 versus needing it at 85. So those 15 years of autonomy, of freedom, that's worth, I believe, a lot. Dr. Lee's team validated this recurrent progression approach using Cox proportional hazards model with the Anderson-Gill method against approved drugs like donanemab and lecanemab.
Cox proportional hazard model with Anderson-Gill method was applied to the time to recurrent progression in for baseline asymptomatic participants in the NTU-001 trial. You can see that the hazard ratio for the time to recurrence is similar to the hazard ratio for time to first progression in lecanemab and donanemab study.
The fact that this new method produces similar hazard ratios, which is a measure of treatment benefit over time, to FDA-approved drugs validates its clinical relevance. It's not just statistical gymnastics, it's capturing something real about how these treatments work over the entire disease trajectory. Next, let me introduce you to Dr. Kunjin from Anavex Life Sciences, who tackled one of the biggest problems in Alzheimer's trials, patient dropout. In very long trials, dropout rates can exceed 30 to 40%, especially in preclinical studies where participants don't feel sick yet. When patients leave trials early, which happens very frequently in these 5 to 7 year study, traditional mixed model repeated measures statistics lose power dramatically. But Dr. Jin developed something called a linear B-spline mixed effect model that can incorporate unscheduled visits and handle dropouts much better by using continuous time modeling instead of discrete visit windows.
The interesting thing is you look in the right column. When you have high dropout, the MR lost power. Here, the new model will increase power, which is a 50% increase. Well, I'm not saying power is still relatively low, but the increase in power is 50%, which means when you have dropout, this model have more room than this model works.
A 50% power increase when dropouts rates are high. This is particularly relevant for us APOE4 carriers because we might have different dropout patterns, either due to faster progression requiring more intensive care, or conversely better response to treatment, making us more likely to continue. But here's something even more innovative.
Here's power. Power with 50% patients have unscheduled visit. One, two unscheduled visit. You can see the yellow is new model. They all have high power than the MMRM, and also more unscheduled visit, you have more power.
So think about this. Every extra doctor visit when you are worried about your memory, every unscheduled checkup when family notices changes, that data can now be used to strengthen the trial rather than just being thrown away. It's using real-world behavior, the natural tendency to seek care when concerned, to improve scientific rigor. Now we come to Dr. Jingling Zhong from Alector, who discovered something fascinating about how disease-modifying treatments actually work mechanistically over time.
Donanemab is an example of disease-modifying treatment. We have the treatment trajectories of CDR over time. We can see that the absolute difference between the two treatment arms increases over time. However, when we take the ratio between the two lines, it is about 40%, quite stable over time. This plot also shows that once the treatment effect sets in, we have consistently nominal significant results from month 6 till the end.
This 40% consistent slowing is revolutionary. It means these amyloid clearing antibodies doesn't just help at one specific point. They continuously slow progression by the same percentage, suggesting they're addressing the underlying disease mechanism rather than just the symptoms. So for us as APOE4 carriers facing much higher risk, a consistent 40% reduction in progression rate could mean the difference between developing symptoms in our 60s versus our 80s, basically buying us 2 decades. That's really not bad. But here's what really matters to patients.
So what is clinically meaningful? Let's consider from a patient's perspective. When there is a stable percentage slowing from month 6 to 18, which the following is more meaningful? Percentage slowing at one time point, such as month 18, or percentage slowing over a time period, such as month 6 to 18. So this is ultimately a question. What do you really want to estimate? So the percentage slowing over a time period may be more clinically meaningful to patients.
Exactly. We don't care about our CDR-SB score at month 18 in isolation. We care about maintaining actual functions over years, being able to manage our finances, remember our grandkids' names, or basically stay independent. And when Dr. Zhong's teams applied this thinking to trial design using the proportional means ratio model,
this slide illustrates the impact of using multiple visit sample size requirement. The 3 plots here provide graphic comparison of power across methods for sample size of 250, 350, or 400 per group. To achieve approximately 80% power, traditional MMRM requires around 400 per group. By using multiple visit MMRM, we can reduce that to about 350 per group, a saving of roughly 12.5%. And using a PMMRM model, the sample size drops even further to about 250 per group, which is a 37.5% reduction.
While a 37.5% reduction is required in sample size by leveraging information across all visits rather than focusing on a single endpoint. Combining this with Dr. Devanarayan's 35% from machine learning's prediction, we're talking about trials that could be 60 to 70% smaller while being more likely to detect real benefits. This makes trials for specific population, well, like APOE4 for carriers who are around like 1 to 2% of the general population, suddenly very feasible. And now here's where the rubber meets the road. These innovations are brilliant, but they mean nothing if the FDA won't accept them for drug approval. That's why the session included 2 FDA representatives, Dr. John Lawrence, acting team leader for neurology drugs, and Dr. Xiu-Jen Wang, the Deputy Director of Biometrics, to provide regulatory perspective. Dr. Lawrence had encouraging news about using these prognostic covariates.
I'd like to point out there's an FDA guidance document on covariate adjustment. It's a very good guidance document. I recommend everybody to read it. So one of the things it says is sponsors can adjust for baseline covariates, and doing so will generally reduce the variability of estimation of treatment effects. So the FDA does encourage covariate adjustment, and the efficiency gains when the covariates are prognostic for the outcome.
The FDA actively encourages these innovations. These guidance documents represent years of regulatory evolution, moving from rigid requirements to more flexible, scientifically driven approaches. But they also provided important warnings about maintaining statistical rigor.
My comments are that the PMMRM, the type 1 error rate, might not be controlled if the model is not correct. It's not even clear how the type 1 error rate is defined. Possibility is that the null hypothesis is that the two groups are the same at all visits, or another possibility is that the null hypothesis is that the two groups are the same at the final visit only. The second type of null hypothesis, it's unclear if the type 1 error rate would still be controlled.
This is the FDA being appropriately cautious about type 1 error, which is the risk of approving drugs that don't actually work, which are basically false positives. They need to ensure we are not just finding statistical artifacts, but the fact that they are actively engaged in these discussions, working through the technical details, is hugely encouraging. And then Dr. Xiu-Jen Wang dropped a bombshell announcement that directly impacts us.
The first thing I wanted to share is that many of you probably don't know that just last month, the FDA added patient selection into the drug label for imaging drugs. The three imaging drugs used to select patient for the clinical trial. And because the result was highly significant, we were able to add information to the label for these three imaging drugs as a drug class update so that these drugs in the future can be used to select patients for clinical trial.
This means amyloid PET imaging agents like flurbetapir, flutemetamol, and florbetaben, which are particularly important for us ApoE4 carriers who accumulate, again, amyloid earlier and more extensively, can now officially be used to select patients for trials. Combined with blood biomarkers like p-tau217, we can identify the right patients at the right biological stage, not just based on symptoms. So let's put all of this together. 6 different research teams have independently developed statistical innovations that converge on the same revolutionary conclusion. Many failed Alzheimer's trials didn't actually fail. We just were not looking at the data the right way. For us as ApoE4 carriers, whether we are heterozygotes facing 3 to 4 times more risk, or homozygotes like me facing 15 to 30 times more risk, these breakthroughs mean, well, first, a drug that failed for the general population might work brilliantly for our specific subgroup, especially given our unique amyloid accumulation patterns. Second, our blood biomarkers, particularly p-tau217, can predict our progression with 83% accuracy, allowing personalized risk assessment. Third, future trials could be 60 to 70% smaller while being more effective, making ApoE4 specific trial economically viable. And this is really important. And fourth, the FDA is actively supporting these innovations with new guidance already implemented. So these are the mathematical foundation for precision medicine in Alzheimer's that we've all been waiting for. It's an era that comes using AI and machine learning, where trials are designed for specific genetic profiles, where every data point from every visit counts, and where we can finally identify which treatments work for which people at which disease stage. A quick word about the Phoenix community. We exist to help ApoE4 carriers like you and I navigate this rapidly evolving landscape. Because when trials designed specifically for us begin recruiting, and they will very soon, we need to be ready and we need to be in contact with these researchers, which is something that we do. We need to understand the science. We need to track our biomarkers and be prepared to participate as informed advocates for our own health. So the statistics presented today aren't just numbers. They are the mathematical proof that precision medicine for Alzheimer's is really possible, it's even inevitable. And for those of us carrying ApoE4, that changes everything we think about the therapy and the innovations that are coming for us and the way we can help in these clinical trials. So keep learning, keep advocating for ApoE4, and remember, we are not just carriers of a risk gene, we are pioneers in precision medicine. Together, we are going to beat the odds.